If you're obviously, I think we've seen these great presentations today from Teresa Lamb and from Ben Murrell. And part of what I think we've all started to appreciate is thanks to vaccination, we are starting to put, at least here in Europe and the United States, and I know there are other people joining us from all over the world, and hopefully in other parts as well, you're starting to hopefully see perhaps light at the end of the tunnel. And this has really been driven by this great global effort for vaccination, both the development of the vaccines, the manufacturing, the distribution, and everything else associated with it. But you perhaps if you pay any attention to the news, you also notice that it seems like almost every few weeks that there's a different headline that can be somewhat alarming. Sometimes which are always related to variants. So they could be the new Lambda variant, perhaps a mutation of Delta known as Delta Plus. There's these highly mutated variants that have emerged out of South Africa, some of them which may have higher or increased transmissibility. There was the Lambda variant associated with escaping potentially vaccine-generated antibodies. Ben did a great job of introducing the Mu variant and some of its potential risks. There's even some guy talking about COVID-22. And then we even have variants that are just been reported as recently as 3 weeks ago. So these headlines keep going on, and if you're like me, you don't necessarily know how to feel about it. Maybe these are all false alarms and they're just meant to get attention, or maybe there's something to it. As we learned from the surge of infections that occurred this summer in certain parts of the world from Delta, we obviously have to be very much concerned about variants and how this could change the trajectory of the pandemic. Regardless of the headlines, one thing that we can certainly be confident of is that the SARS-CoV-2 virus will continually evolve, and that the emergence of new variants is all but guaranteed. And if, of course, as you've seen here in this phylogeny, that by starting simply in the corner here in December 2019, that we've seen all of these very large clusters of mutations and variants that have emerged, including, of course, the ones that are highlighted here, like the kappa and the lambda and the beta and gamma, and of course, delta, and how much it has become dominant across the globe. As we know now, many of the variants have mutations across, and Ben did a great job of describing some of the various parts of the spike protein that are associated with mutations. And it's oftentimes mutations in these spike proteins that are an important part of these variants and the mutations that are associated with these variants. Now, one of the things that my research group, along with collaboration with several others, developed, and I don't have a lot of time at all today to talk to you about it, so I just only point you to this preprint that we've put online, is essentially a method to perform simultaneous diagnostic detection and genomic surveillance of SARS-CoV-2. And this was a great effort with collaboration with a lot of researchers who are all listed here across different groups, both within Switzerland and globally. And in this methodology, what we developed is an approach to basically extract swab or saliva samples. And instead of doing the traditional PCR tests, what we do is we actually perform a test that's based on deep sequencing. And it relies on doing something where you add molecular barcodes. And by adding molecular barcodes, you basically are able to tag patient samples. And you're able to perform what we call multiplexed PCR. And this allows you to not only to target more than just the 2 sites of the virus, which are typically just what are done in these normal standard PCR tests. So we have many sites of the virus that we target, and then we perform this next generation sequencing or deep sequencing assay, And from that information, we're able to not only detect viruses based by the sequencing reads, we can also perform the genomic surveillance, which is basically identify mutations and perhaps even classify variants. And we have applied this method on samples, on control samples that show you that we can detect virus at very low numbers, copy numbers, as well as the correlation of this with the standard PCR tests is very good, and that we can cover mutations that are across the spike protein, including that receptor binding domain that is often associated with mutations in some of these most notable variants of concern. So I point you towards that preprint just to acknowledge the really great work from all the researchers listed here that contributed to it. Now moving forward, I want to now talk again a little bit more about the receptor binding domain that Benjamin also already introduced. As you see in this picture here, the RBD is this dark gray structure is listed here, and it's engaging with this host ACE2 protein here. Of course, ACE2 is the protein in which the SARS-CoV-2 virus engages with and uses for entry into host cells. And it turns out that the RBD and the sites in which it engages with ACE2 is also one of the prominent sites for which neutralizing antibodies target. So these are actually 4 clinically approved antibodies that are either from the company Regeneron or Eli Lilly, which have been approved and have been used as therapeutics. And these neutralizing antibodies target the RBD, and by and large part, they essentially block its ability to engage ACE2, and that's its primary mechanism. These antibodies, their primary mechanism from neutralizing viral infection. Now, thanks to a lot of research from many groups to study antibody responses against the RBD, we've essentially learned that if, again, here now the RBD is in this gray structure, that there are actually, the neutralizing antibodies can be broadly categorized into 4 classes, which are listed here. Now, these classes are meant to represent the site at which the antibodies target the RBD and essentially inhibit ACE2 binding. And so you can see here that there are class 1, 2, 3, and 4, and that some of the antibodies listed here in class 1, like the Regeneron 10933, is one of these clinically approved therapeutics. CB6 is one of the clinically approved Regeneron 10987 is one of the clinically approved antibodies as well, and so is CB6. And so what these neutralizing antibodies and these classes represent is they're meant to basically be an overall representation of the types of antibodies we would make in human immune responses. These could be responses to natural infection, but they can also be vaccine-induced antibodies. And if we now look on the right side here at the spike protein, and a close-up here now is of the receptor binding domain, and mutations of these variants of concern, which are listed here. And what you start to see is there are actually a lot of the variants possess some common mutations. One of the ones that's included here is the N501Y mutation, which was present in the alpha variant, and then has also been present in the beta and gamma variants. This mutation is associated with enhanced binding to ACE2. There are also mutations in position 484 and 417 of the RBD domain, which has also been associated with escape from neutralizing antibodies. In fact, it appears as though the gamma and beta variants, which are very similar in terms of their mutations, may have had antibody escape as one of the selective pressures that drove them to becoming dominant variants, at least at the time when they emerged in early 2021. So if we think about variants, they will continue to develop as we go on. And if we think about the number of variants that just will, of course, grow over time, which is here in this blue curve, what that also means is that eventually the antibodies, that we currently have will eventually start to escape, the variants will escape these antibodies. Kind of a good example of this is that in November 2020, there was a sort of landmark achievement in that Eli Lilly had the first COVID-19 antibody therapeutic clinically approved. So that was in November of 2020. By April of 2021, just a few months later, the FDA revoked the approval of this drug. The reason why, of course, was because new variants had emerged, notably ones like beta and gamma, as well as delta, that could escape this antibody almost completely. And as Teresa Lam mentioned earlier today in her presentation, we do know that the antibody neutralization to some of these variants is reduced. Now, it doesn't mean that this is data from vaccine efficacy of somebody receiving the Pfizer BioNTech vaccine. So we do know, of course, the neutralization to one of the original versions of the virus is extremely strong, but we do see there are examples where there is reduced neutralization, like the mu variant and the beta variant. So we do have to appreciate that eventually mutations will emerge that can escape some of these antibodies and potentially show partial resistance to vaccines. Now, to understand the impact of mutations to the RBD and its escape from antibodies, a method called deep mutational scanning was applied. And this has largely been driven by a researcher named Jesse Bloom, who's at the Fred Hutchinson Cancer Center in Seattle and in the United States. And what he developed was a yeast display system where he expresses on the surface of yeast a mutated library of the RBD domain. And what he does is he makes single point mutations to the RBD all across the entire RBD, which is about 200 amino acids long. So he has this large mutagenesis library of about 4,000 variants, all with one mutation tiled across the RBD, and then he screens it experimentally by flow cytometry to identify antibodies that bind or escape to these mutations. And then uses deep sequencing as a readout. And what he can start to identify is essentially mutations that escape from, say, this is a clinically approved antibody called LICO-555. And here's another therapeutic antibody. And he can identify the mutations that escape those antibodies. And of course, he can compare those, they can compare that with variants that emerge naturally. And then their research also is able to build escape maps that look like this. Now, Jesse Bloom and his research group have been very prolific, as you can see by his publication record just in the last year and a half, which shows you the impact this method has had on the field. But one of the things that one has to appreciate is that variants are emerging that have more than one mutation in the RBD. And in fact, multiple combinatorial mutations in the RBD are all but inevitable now. We've seen this with the beta and gamma variants, but also this recently reported variant called C12 out of South Africa has 4 mutations in the RBD alone. So what we need to understand is that what might be very powerful is if there is a way to predict the impact of combinations of mutations in the RBD. And in particular, how that would impact binding to ACE2 or escape from antibodies. This would, of course, provide us with a means to essentially have a better idea as to how, threatening such a variant could be to potential resistance to immunity or vaccination. So in order to do this, we developed a method that we call deep mutational learning, which allows us to perform predictive profiling. So what we did is we first took the RBD domain, and as you see here, it's about 200 amino acids characterized by position 333 to 527 of the spike protein. But there are particular sites of the RBD that are mutating more often based on the natural genomic sequence data. and that are also engaging with the ACE2. Sometimes these are called the receptor binding motif, or RBM here. And so we selected sites within this RBD domain, most notably 484 to 505, as well as position 417 and 439. And what we did is we created a mutagenesis library in yeast, similar to the work of Jesse Bloom, and then performed flow cytometry to sort first variants and mutations that maintain binding to ACE2 or escape binding to ACE2. And then we took the ACE2 binding fraction and we screened them for binding or escape to the 4 clinically approved antibodies listed here, which represent the three of the 4 classes. And you can see here is then the flow cytometry for that. And then when this data was retrieved, so once we do all this sorting, we then perform deep sequencing on all of the populations. And we use the deep sequencing data to then train machine learning models. And the goal of the machine learning model is to basically then take a sequence, any of the potential hypothetical mutations that exist, and make a prediction on whether they bind to ACE2 or escape on a particular one of these antibodies. And eventually this allows you to build these very information-rich data sets, which I will explain to you a lot more in the next few minutes. So the first thing that we had to do was design and then screen a mutagenesis library of the RBD. What's pictured here is the position 484 to 505 that I mentioned. And what we did is we actually took some of the data from Jesse Bloom's group to help us design a combinatorial mutagenesis library. So what this means is that there are multiple mutations, obviously, and they are tiled, or they're made across this region. We keep some positions fixed, as you see, like for example, 486 has an F. The reason why is that the data from Jesse Bloom indicated that if you make any mutation to position 486, you lose all binding to ACE2. So we don't really learn a lot from if we don't have any ACE2 binders. So these are why we keep some positions fixed. Now, overall, this combinatorial mutagenesis library is greater than 10 to the 12. So it's a massive, trillions of possible sequences, and that is far more than what we can experimentally screen. Experimentally with yeast display, the most you can screen is about 10 to the 9. So several orders of magnitude lower than the theoretical diversity. We also created what we call a low distance library by using tiled mutagenesis. So now what we do is we only mutate 3 positions, and we tile this across, so we end up making 220 different oligonucleotides that create these low distance mutagenesis libraries. And this also is an important step in how we are able to create both high distance and low distance mutational data. So all of these experimental libraries are transformed and expressed in yeast, and then again by flow cytometry, we then sort populations that maintain binding to ACE2 or escape ACE2 binding, and the same for these clinically approved antibodies pictured here. And then this is the output of the sequencing data, and what I hope that you can see here is that while there is some shift between the binding and non-binding patterns, overall, it is very difficult to, by eye, identify any really strong motifs. And that is really where the power of machine learning will lie. It will be able, if you can train an effective machine learning model, to look at these sequences and essentially identify very complex patterns that allow it to identify whether a sequence is either a binder or a non-binder. So getting to that, we created this data set that crosses the different distances of possible mutations. And what happens is you then take these data sets, which consist of, remember, of binding and non-binding, and these sequences are encoded in a very simple approach called one-hot encoding. So these are now, remember, amino acid sequences of the RBD domain that then we turn into this matrix so that we can actually input it. And then we train different types of machine learning models I will show you a little bit of how I won't have time to really explain all of the benchmarking that we did, but eventually we decided to use a random forest and a recurrent neural network that's pictured here. We compared many different both simple and complex models, and both of these seem to perform the best. And then what happens is both of these models are used, and so for any given sequence, what we would aim to do is make a prediction on whether that sequence binds ACE2 or doesn't bind ACE2, and the same for antibodies. And then if both models agree, we have a much stronger confidence that that is a true ACE2 binder or a true antibody escape variant. Okay, so once we went through this process, we of course did the deep sequencing, and we did a lot of data processing that I don't have time to share with you today, but then we could assess, though, the performance of our models once they're fully trained and optimized. And one of the most common ways to assess a model is using something called the receiver operating characteristic curve, which is just a way where you plot true positive and false positives. And what essentially you want to look for is that a really bad model is here on this diagonal line or in green, where basically it's more like a random guess because you have as many true positives as false positives. A really good model is moving towards the red or the blue, where you have so many true positives with a very low false positive rate. So when we trained our ACE2 binding model, sorry that the colors are difficult to see, but we have diagonal lines which represent the controls, which we actually just shuffled the data and checked if the model could learn anything, and indeed it didn't. But when you have the correctly labeled models data, you can see here, these are the random forest and the LSTM RNN performing extremely good. We use an area under the curve as an example, and the area under the curve here being close to 0.96 and 0.97 for both models mean that they're extremely well performing. Similar results were made for each of the 4 monoclonal antibodies that we tested. So remember that essentially we create a machine learning model for every antibody, and then again checked for their performance, and you can see that we have extremely good models for predicting antibody binding or escape. So just to give you now an example of why deep mutational learning is powerful, if we look at deep mutational scanning data published from Jesse Bloom's group in this study from earlier this year, they took the antibody Regeneron 10933, and they don't really identify escape for E484 mutation, and they don't identify any escape mutations in position 490 because it's not listed here in their escape map. Now, it turns out that when we can apply deep mutational learning, and we can check our random forest model written on top here and our RNN model on the bottom. And the probability here is just saying the probability that Regeneron 10933 maintains binding to this mutation. So very similar and correctly corroborating with Jesse Bloom's data, we see that for both models that E484K is not an escape variant of Regeneron 10933, neither is F490I as the example here. So that's good. Our data corroborates that of DMS and Jesse Bloom, but one of the really powerful things that our deep mutational learning can do that cannot be done with deep mutational scanning is that we can look at the combination of these 2 mutations. And now you can see they actually the probability of binding has dropped dramatically, and these are actually predicted to be escape variants. So this shows you just how it's powerful. And of course, we can look much beyond just 2 mutations. And so that is what we next did. So here is what you're looking at is essentially a synthetic lineage. So all of these are sequences. So we are able, remember now with deep mutational learning, to predict across hundreds of millions of possible variants. That's what our model is able to do. And so we decided to select 46 sequences that we just synthetically more or less generated and asked the model to make predictions on. And these are coming from, this is the alpha variant, and then here is the E484K mutation, the E484Q, which I believe is part of the kappa variant, and then additional mutations 3, 5, 7 away from the original virus. And then here is what our model predicts, either ACE2 binding or, ACE2 non-binding in red or ACE2 binding in green. And what we find is that the X's are the only predictions that were incorrect. So 43 out of the 46 sequences, we expressed and tested all of these physically. So these are all on the left, in silico predictions by the machine learning models. And then on the right is the actual experimental data, and we were correct 93% of the time for ACE2 binding. So you can see the model is extremely good at predicting ACE2 binding. Of course, we also then checked to see if our predictions for antibody escape were correct. So these are the 4 antibodies. And so we tested the 36 variants that maintain binding to ACE2. And for the antibody predictions, we were correct altogether. Again, 93%, 133 out of the 144 predictions were accurate. Some of the antibodies were extremely accurate, like the Regeneron antibodies. So that shows you again how we're able to predict escape. And if you think about it, we're kind of around here. This is like the beta variant potentially, or similar to the beta variant, about 2 mutations away. But what we identified, which is of concern, is that you only require 2 mutations away from alpha to identify variants that were predicted to escape all 4 neutralizing antibodies. And this is the mean fluorescence intensity, meaning that we expressed physically these 4 antibodies, these 3 variants, and tested them. And indeed, they all escaped the 4 antibodies. So it really shows you that there is the potential to only make a few more mutations and escape more antibodies, which is certainly a concern. So another thing that we did that was kind of fun is that we combined this deep mutational learning with AlphaFold. So if you just very briefly, AlphaFold is this, really groundbreaking development by Google and DeepMind, and they partnered with the EMBL EBI to create this protein structure database. And AlphaFold is an AI-driven system to predict protein 3D structures when you have sequencing data. And they have this landmark paper that was published earlier this summer. So we used AlphaFold to predict the structures of some of our variants. And so if you take a look here, the black box here is the original Wuhan virus and the AlphaFold prediction of its structure. And then if you actually look at some of these variants that are following this lineage here, which we've experimentally tested all of these, including, so these are the DML predictions, but they also experimentally are confirmed, you can see that there are variants here in green that maintain ACE2 binding, but have an extremely different looking structure than the wild type. So why is that important? It's of serious concern because it means that SARS-CoV-2's RBD can undergo massive structural changes and completely have this unique diversity, but still maintain binding to ACE2. So it means there's a lot of evolutionary landscape for it to explore and will probably still maintain ACE2 binding, which means it could still maintain potential infection. Now, very briefly, I want to just, because I know I'm running out of time, I just want to hit one of the final points. So if you look at the way that we profile variants, a great example is Stanford University developed this database, which all they really did is they curated a lot of information that is available from many different studies. And they listed on top here are all of the different, or many of the clinically approved antibodies. They use different names here. Bamlanimab, for example, is LICO55, and CAS is Regeneron 10933, et cetera. And then they took some of the well-defined most notable variants, and then some of the most notable mutations, and they compiled the experimental data. And you can see basically in blue means that the beta variant completely escapes this antibody. Now, the thing is, they've done this on these, small number of sequences, and they did this retrospectively after we knew the variants were important, the mutations were important, and then of course it required a lot of, experimental effort to do this. Now, the real power of deep mutational learning is that instead of doing this retrospectively, we can actually do this prospectively. Because we have this data set already in these machine learning models, we can literally type in every sequence variant that's one distance away from alpha, and that maintains ACE2 binding and ask if it escapes from one of these 4 neutralizing antibodies. And this heat map shows the 2 different types of models for the 4 different antibodies. And you can see in most cases, the 2 models agree with each other. And so we can see that it only takes, one mutation away from alpha to escape some of these antibodies. And then there are examples where just 2 mutations away from the alpha variant actually escape 2 or 3 antibodies. We can do the same for any lineage, so we can just type in the beta variant lineage and follow its trajectory. And then, of course, what's a lot more concern is you can see 1 mutation away from beta starts to escape potentially 3 antibodies, and 2 mutations, there's examples of escape of all 4, similar for kappa as well. Another thing that we can do is build what are called deep escape networks, which are very complicated data visualization here. But basically, what it's showing here is that everything within the circle is 1 mutation away from alpha, and everything outside of the circle is 2 mutations away from alpha. And here is the escape of the number of these clinical antibodies. And you can see that essentially there's a small number of 2 mutations that will escape all 4 antibodies, but there's quite a few mutation variants that will escape 1, 2, and 3. The situation, of course, looks much worse with the beta variant, where it takes 55% of the variants that have 2 mutations would escape 3 of the 4 clinical antibodies. And of course, you can apply this other way. So this is a really great way of just identifying potential evolutionary trajectories. And then the final example of how powerful this approach could be is that if we think about all these headlines, what we can now do is you can see this headline from a couple of weeks ago about the new B11523 variant, capable of escaping immune protections. So what we could do is we could simply go in, we looked it up that the B11523 variant has these 2 mutations in the RBD. Deep mutational learning predicted that it would escape Li55, okay? So you can see that here because Li55 is this third panel, and every single 1 mutation variant escapes it. So that's correct. That's what we expected. But now you can also start to see if B11523 ever became a variant of concern, you could start to see the trajectory and how much should we be afraid of it. Clearly, we should be pretty afraid of it because by the time you get 2 mutations away from it, you can escape a lot of antibodies. And so we don't have to wait and do experiments here. We can just type this in because, of course, we only saw that headline from 3 weeks ago, and then we can generate the data basically overnight. And again, we can do escape maps as well as other types of data analyses. Great. So just to summarize, hopefully I've shown you that deep mutational learning can be very powerful for profiling SARS-CoV-2. It allows us to really see that the highly diverse landscape that basically the SARS-CoV-2 virus RBD domain has to maintain binding to ACE2. It enables prospective profiling of antibody escape. Now, in the future, we think we could, if SARS-CoV-2 antibody therapies need to continue for many years, there may be a way to use DML to help us design the optimal combination of antibodies that cover the most potential mutational variants. And then we also think that if you start to screen this against large panels of antibodies, perhaps say polyclonal antibodies from vaccinated serum, you might be able to identify variants that have the most probability to show some vaccine resistance. This may allow you to start putting some likelihood attached to potential future variants that could emerge. So with that, I will just thank a really great group of researchers in my lab that led this study. We worked very closely with a spin-off company, DeepCDR, and Cedric Weber, who was part of this. We have, of course, a lot of great other team members that were involved in directly or indirectly, and obviously the BRCCH for their support and funding and all of the great opportunities that they've enabled.